Father, and of the Son, and of the Holy Spirit. Amen. Lord, grant us concentration, that we may learn, listen, understand, and have a peaceful mind, and remember that you're with us always. Amen. So we've covered the first two types of tissues in plants, the dermal tissues, the outer layer, and the ground tissues, the photosynthetic, the storage, the support cells. Now we're going to begin talking about the last set, which are the vascular tissues. So the first thing that I want to indicate to you is that we divide the vascular tissues into two types. The xylem, which we will talk about first, and then the phloem. An important note to start with is their location in the plant. And you'll notice that the xylem are more centrally located, closer to the center of the stem, the root, whichever part of the plant we are, whereas the phloem is located more peripherally, closer to the ground tissue and the dermal tissue. And we're going to see that's an important consideration when we talk about how these particular vascular tissues differ. Okay, so we're going to start with the xylem. The xylem are going to be, again, centermost part of the vascular tissue. They are going to primarily be conduits for water, although water will contain ions, dissolved minerals in there, so as they're moving the water, they will only move water and dissolved minerals, ions. Generally speaking, and I can say this a little more confidently than that maybe, but they're only going to move in one direction, and that is up. So the movement of water in the xylem is only going to go from the roots up through the stem to the end of the plant. And we're going to talk more about that process and its relationship to water potential in a little bit. Another note that's important to make with relationship to the xylem is that they do not contain living cells. It's really cell wall remnants is what we have to be able to carry the liquid through. So these do not contain living protoplast, similar to what we talked about when we talked about the sclerenchyma cells that were present in the ground tissues. So for the vessels themselves that make up this, or the xylem, there are two different types. There are vessels, okay, and this is showing the vessel here, okay. And so the vessels are larger cells, they are shorter cells, they are wider, they have a larger carrying capacity. Because they're wider, we always should connect that together, they are going to be shorter distance that it's going to carry the water up through there. They are also cylinders, okay, and as we're going to see, we're going to contrast this with the tracheids, okay, which have sort of an angular end on it. The vessels are going to be larger and they're going to be flat on each end. The other type, the smaller type, as we can see in this picture here, okay, the smaller type are the tracheids, okay, and the tracheids are longer and thinner. Obviously, they will contain a lower volume of water that they can contain, but they're not completely cylindrical. They're going to have sort of these tapered ends that are going to be present on the cells. Okay. And so these are going to be longer. They can move farther distances, the cells themselves, or maybe I should say the former cells themselves, because these two are only cell wall remnants, and they're going to be able to carry the water up through the plant. So that's the xylem, more centrally located, carries water and minerals, and contains these two types. The second type of vascular tissue is the phloem. And the phloem unlike the xylem, okay, has two unique characteristics. First of all, the phloem, unlike the xylem, carries more than just water. Okay? The phloem is going to be the location where we move the chemicals. Okay? This is going to be where the sugars are carried, hormones are carried there too. So the phloem is going to contain the sap. So you may have again seen some of the buckets on campus that were collecting the maple sap that would be boiled down to make maple syrup. In this case, that's going to be carried in the phloem, which again makes it nice because they're more peripherally located, closer to the dermal cells, which means it's easier to access them. The other difference with the phloem is that it's got bidirectional movement. Okay? It can move the fluids, it can move the sugars, the hormones, both up the plant, but it can also move down the plant. So in the spring now, when we're moving sap up the tree to provide the nutrients to start growing the leaves, in that case, it's primarily moving up the tree. In other times of the year when we're storing it in the roots in that, the phloem's able to carry down to the bottom. Okay? And I've already mentioned this a couple times, but it's more peripherally located, closer to the outside of the plant, closer to the dermal tissues. Now, unlike the xylem, the phloem do contain living protoplasts. Okay? And in fact, we're going to see that there are two different types, a sieve tube member and a companion cell, that are going to make up the process or make up the organization of the phloem. And so the sieve cell or sieve tube member cell are elongated cells which will make up the tubes which will actually carry the fluid. Again, it can go in both directions depending on the loading, as we'll talk about in a little bit. So these are going to be separated by plates. Again, that provides some of the function as far as the ability to move. The other type of cell, the companion cells that are present there, are actually ground tissue cells. So again, location is important. They're going to be located closer to the ground, closer to the dermal tissues, and so they're going to have this support cell that's associated with them. And as we'll see in a little bit, we're going to see that these companion cells are going to be important in phloem loading, in the process by which the phloem is able to move in both directions. So one side note before we talk about the mechanisms of water transport, and that is the organization of the vessels themselves, of the vascular tissues. So we're going to talk a little bit more about this in a little bit, but flowering plants we can divide into two different types. Okay? They'll either be the monocots or monocotyledon, okay? And that's what the cot stands for. Okay? The monocotyledons, this should be an O, okay? Monocotyledons means they have one embryonic leaf. Okay? And we're going to talk about some of the other features of these. In this case, the vascular tissues are not specifically organized, but they are randomly found throughout the plant itself in these vascular bundles. Okay. So we have a cross-section of a stem here that shows the vascular tissue sort of in random locations throughout the ground tissue. One of the notes I would make only, if we looked at a close-up of one of these, we would still see the same sort of orientation, where closer to the periphery is the phloem, where the xylem is more centrally located. And so even though we don't have a significant organization of these vascular bundles, they are going to show a similar as far as the location goes. Now, dicots or eudicots, true dicots, if you will, okay, again, the cot here is cotyledon, means that they have two embryonic leaves, or we could say two cotyledons. In this case, the vascular tissue is not randomly positioned throughout the ground tissue, but it's found in this sort of circular pattern around the outside or center ground tissue. Okay? And so this is going to have a particular function or particular relationship to growth. So the vascular tissue forming this round structure or circular structure around the outside, where again the phloem is the outermost and the xylem is going to be the innermost layer of the vascular tissue. The importance of this organization is that it allows for secondary growth. In other words, when we talked before about the meristem cells being located in a lateral position to allow for secondary growth, part of the reason they're able to do this is because of this ring arrangement of the vascular tissues. So this gives you a little preview. We're going to come back to the monocots and dicots in a little bit and talk more about them. What I want to start right now is talking a little bit about the transport mechanism. Okay? So when we talk about the transport mechanism of water, the first note that's worth making is that the xylem will only carry water up the plant, whereas the phloem carries carbohydrates and nutrients in both directions. Okay? It's not limited to move simply up the plants. And so we can talk about two different types of movements in this. We could talk about the pushing and the pulling of water. And the latter is probably the more important of the two, but let's start with the pushing. So the pushing of water is occurring at the roots. And so what can happen is if there's a lot of water in the soil, that water and minerals can be pushed in to the roots to sort of begin the process of water movement. So the water will get in, but it still has to move somewhere. The more important process is the pulling or transpiration. Okay? And what is transpiration? Well, think of it this way. If we were to look at the water that's inside the xylem, we would see there are two actions or two processes that are present. We have adhesion, in which the water is going to be sort of connected to the inside layer of the cell walls that make up the xylem. So adhesion is sort of connecting it to there. But more importantly is cohesion, which is holding the water molecules together. And so when you see the water molecules that are present, for example, in this top image along the leaf, they're going to be connected together by this process of cohesion. Remember, hydrogen bonding that will bond the water molecules together. Okay, and so here's where the process gets interesting. Okay, you're probably already aware that what I just described to you is a little more detailed form of capillary action. In that if you have a straw in water, the straw or the water will start to climb up that straw depending on how thick it is. Well, the second part of this is going to be evaporation that's occurring at the stoma, at the openings in the leaf. So as the water evaporates out of the leaf, out of the stoma, okay, it's going to sort of pull the water up. Think of those as beads that are sort of attached together. And so it sort of pulls it up. So in the process of pulling at the leaves, it's going to pull up here, it pulls up here, it pulls up here, and it pulls up there. So we have this constant pull that leads to evaporation and release of some of the water into the environment. Okay, we'll talk about water potential in a little bit. But that evaporation is what leads to the pulling of the water through there. And this pulling is going to move the water through the xylem up through the, in this case, up through the tree. As it's moving up, the water can be removed at different points if it's needed by the cells in that location. So this is the process of transpiration. To really understand how transpiration is working, we need to talk a little bit about water potential. So what is water potential? Well, water potential is the prediction on the movement of water in a system based on physical pressure and solute concentration. Okay, so the prediction on the movement of water based on physical pressure and solute concentration. And so we can calculate that based on this equation. The water potential, or psi W, equals the physical pressure, or pressure potential, psi P, plus the solute potential, psi S. And so water, as I'm sure you're aware of, okay, will always move to lower physical pressure and it will always move to a higher concentration, okay? Or we could say a higher solute potential. Okay. And so how does this work, or how can we explain this? Well, we can explain it this way. If we break it down into the two different parts, physical potential and solute potential, okay? If we have water inside the cell, which is exerting pressure against the cell wall, and lower pressure exerting into the cell wall, this means the net movement of water will be out of the cell, okay? Simply because there's more pressure. Again, think about, use the balloon analogy with the guard cells. Think about a balloon. If you put more pressure on a balloon, that's going to expand the balloon as that physical pressure pushes out the sides of the balloon. Same idea here. The physical pressure inside is going to cause the cell to swell against the cell wall. Okay. Now, solute potential, on the other hand, is going to be determined by the concentration. And so the higher the concentration, the lower the solute potential. Okay? That's important to keep in mind. Water will always move towards a lower potential. And so in the case of the cell we're showing here, okay, we have a minus 2 megapascal solute potential outside the cell and a minus 0.7 megapascal water potential inside the cell. So in this case, water is being drawn into the cell simply because the water potential or the solute potential is lower inside the cell. So water will always move from a more positive to a more negative potential. When we talk about the movement in plants, it really comes down to the fact that we're really talking about the solute potential as being the most important factor in determining the movement. Okay? And so an example of this, when we talk about water potential in plants, we can see that in the movement, as we'll see in a minute here, but we can also see this in what's called turgor pressure. And what turgor pressure is simply is how much force, again this would be physical force, is the cell exerting against the cell wall. So in cells that the central vacuole is very full of water, that means that there's going to be a high water potential against the cell wall, and so they're going to maintain pressure against the cell wall. Now imagine you're not very good at taking care of your plants, and after a couple of days of not watering, what happens? Well, it probably looks something like this. The leaves begin to droop, they begin to sort of wrinkle up and lose pressure. What you're seeing in this case is plasmolysis. And plasmolysis is the loss of water, which results in the shrinking of the cell. Okay? This is plasmolysis occurring here, where the loss of water has caused the cell to shrink inside the cell wall, and so there's no longer pressure against it, which means the plant is wilting. And so that's going to be a good example of how water potential is used in a cell. If you water the plant because the concentration is high inside the cell when it's in plasmolysis, that water will rush into the cells and cause turgor pressure then to sort of reinflate the cell. Okay. Just a side note here real quick is remember that the movement of water, some water can sort of move through the membrane. It's slow movement and it's not very much. Most of the water is going to be moved through the process of facilitated diffusion, using aquaporins, using proteins in the membrane to facilitate the movement of water. And so opening and closing aquaporins can lead to changes in the movement of water. So the last note I want to make in this video is going to be the relationship between water potential and transpiration. Okay? And so what we're looking at here is a tree, again, same tree we were seeing before. On the side now we're showing water potential, and notice it's getting more negative as it moves that way, as we move up the plant into the air. Okay? And so the first thing you should note is that the air itself has a very high negative water potential, which means it's very likely that water will move from the plant to the air. You should also note that the soil itself has a water potential near zero. So inside the plant is lower. So the movement of water in, whether it's through the membrane or through an aquaporin that we're moving in, is going to be facilitating the movement. Once the water begins to move up, you notice we're moving from a more positive to more negative, so water is going to move up even though it's against gravity. When we get to the stoma and we open that up to the air, water very rapidly will be pulled out. And so the cohesion or the connection between the water molecules will allow it to be pulled up through the vascular tissue. I'm going to stop this video at this point in time. Okay, we're not quite done with water potential yet. We want to talk about one other factor that relates to that. But I'd like to end here and pick that up with the next video. So if you have questions, as always, post those questions on the Schoology discussion board, and I will get back to you with an answer. Thank you for watching.